Everything you need to know for GCSE Biology Paper 1, Part 2. If you haven't watched the other part, go and watch it. Starting off with the cell cycle, which begins with replication of DNA and organelles. And this must happen because the normal body cell contains 46 chromosomes. And we want to end up with 2 genetically identical daughter cells, both containing a diploid number of chromosomes, both containing 46 chromosomes. So for that to happen, we must first double that 46 into 92, which can then go through the stages of mitosis to split into 2 sets of 46. And of course, we require double the number of subcellular structures as well. So your mitochondria, your ribosomes, your cytoplasm must all duplicate as well during this growth stage, otherwise known as interphase. And after this growth phase, we have mitosis, which is made up of separate stages, which we'll go through in a moment, and then the cell divides as the final part of the cell cycle. So we now have double the number of chromosomes, the growth phase has already happened, and we begin talking about mitosis. And the first stage of mitosis is these chromosomes condense, they become visible, and they line up at the equator of the cell or the center of the cell. When I say that the chromosomes condense, this means that the nuclear membrane has broken down and the chromosomes become visible. They're no longer in this densely packed nucleus. Once these chromosomes have lined up at the center of the cell, the equator, spindle fibers form and they attach to one half of the chromosome called a chromatid. And they start to pull on these chromatids. This results in the chromosomes being pulled apart into the sister chromatids and one half of the chromatids going to one side of the cell and the other half being pulled to the other side of the cell. Now a new nuclear membrane can form around this set of chromosomes and this set of chromosomes, creating 2 nuclei, and that ends mitosis. The cells don't yet split, they're ready to split. And it is after mitosis, the last stage of the cell cycle, that the cytoplasm and cell membrane split to form 2 genetically identical daughter cells. So we started off with 46 chromosomes, we doubled to 92 during the growth phase. These then started dividing up, splitting back into 2 sets of 46 chromosomes, formed 2 genetically identical nuclei, each containing a diploid number of chromosomes 46, and then after mitosis finished, cytokinesis occurred, which is where the cytoplasm and cell membrane split. Overall, I want you all to remember that mitosis occurs for growth and repair of body cells, and I want you to remember the stages of mitosis specifically, and growth and cytokinesis being part of the cell cycle but not part of mitosis. Mitosis was just where the chromosomes condensed, they lined up at the equator, they got pulled apart by spindle fibers to opposing parts of the cell, and then 2 nuclear membranes formed around the 2 new sets of chromosomes. For Edexcel, these stages are called interphase, prophase, metaphase, anaphase, telophase, and then cytokinesis. For AQA, you just need to remember what happens within each stage rather than the names. Next, looking at stem cells, human stem cells include embryonic stem cells from embryos and adult stem cells from, you guessed it, adults. Now, embryonic stem cells are found on the inside layer of an embryo. They're unspecialized, undifferentiated, and they can differentiate into any type of cell. Whereas adult stem cells are found in the bone marrow, skin, liver, brain, umbilical cord, blood, but they have limited differentiating opportunities. They can only differentiate into a limited number of cells. This means that embryonic stem cells are extremely powerful in medicine because they can differentiate into any organ cell, into any tissue cell, whereas adult stem cells are very limited. They usually turn into some skin cells, some blood cells as well, white blood cells, red blood cells. And the reason why stem cells are so crucial in medicine is because they've been used to treat some really serious things. For example, diabetes can be treated using stem cells by using those stem cells to produce insulin producing pancreatic cells and then transplanting them into the pancreas of someone who's a diabetic to help them start producing insulin again. Paralysis has also been treated using stem cells by growing nerve cells, neurons, and replacing damaged neurons, damaged nerve cells in somebody who's paralyzed, and therefore replacing those damaged nerve cells, allowing for nerve function to be restored. So the pros are pretty clear. There's a great opportunity to treat diseases from diabetes to paralysis. Adult stem cells are already being used successfully to treat these, and organs developed from a patient's own stem cells are less likely to be rejected by the person when they're transplanted. The issue surrounding stem cells is when they're cultured inside a lab, they could be infected with a virus, and then that virus could be transplanted into a human. Not so good. There is a risk of stem cells also accumulating mutations, and then those mutations leading to things like cancer potentially. And there's a very low number of stem cell donors who are willing to give up their stem cells for other people's treatment. Social issues could include the fact that it's possible for embryonic stem cells to be extracted from the amniotic fluid or the umbilical cord blood after birth, but this is a very expensive option and therefore not accessible to all. And educating the public sufficiently about all of these things that stem cells can be used for and cannot be used for is very difficult. And of course, you may be asked about ethical issues. Stem cells sourced from unused embryos produced in IVF treatment. Is that ethical? Is it right to use them? Who gives permission to use them? Another ethical issue is it's possible to produce embryos through therapeutic cloning, then destroy the embryo just after harvesting those stem cells. Is that an ethical thing to do? And should an embryo be treated as a person with human rights or as a commodity? These are all things you should discuss in your exam. Moving on to the 3 types of transport. Diffusion is a passive form of transport. It does not require any energy, and it moves particles from a high to low concentration down a concentration gradient. Osmosis is a special type of diffusion for water molecules, and it's where water moves from a high to low concentration, more specifically from an area where it's dilute to an area where it's concentrated. Active transport is the only one that goes against the concentration gradient, which means it requires energy from respiration in the form of ATP. So it's an active form of transport, and it moves particles from an area of low concentration to an area of high concentration. One thing we must remember to mention for osmosis is that this is across a partially permeable membrane as well. You also need to be able to talk about the factors affecting diffusion. Firstly, temperature. The higher the temperature, the more kinetic energy, the more movement energy those particles have, the more likely they are to diffuse across. Secondly, diffusion distance. The shorter the diffusion distance, the greater the rate of diffusion, because the less the distance that the particles have to travel, the faster they can travel that distance. Third, concentration gradient. The steeper the concentration gradient, the greater the rate of diffusion. So you can see here, loads of red particles on this side, very few particles on this side, so concentration gradient is quite high, therefore diffusion rate is quite high. If I was to only have 1 dot on this side, there's 3 at the moment, then the concentration gradient would be even steeper and the diffusion rate would be even higher. And finally, surface area, and we're talking about the surface area across which the particles have to diffuse. So the greater the surface area, the greater the rate of diffusion, because there's a greater area over which diffusion can occur, therefore the rate is higher. And that's part 2. My name was Chris, and make sure you let me know what other topics you want me to cover before your exam tomorrow, and send this to a friend so that they can also revise this before their exam. And drop a follow.